Hello, my name is Tatiana Kuzmenko. I'm one of the instructors who teaches General Biology Laboratory at Loyola Marymount University. I'm excited to present our plant-derived drug discovery inquiry-based laboratory module that we developed to introduce undergraduate students to the lab environment, as well as spark their enthusiasm for plants by emphasizing their medicinal aspect. This project cuts across disciplines of plant biology, physiology, and chemistry, providing students with opportunities to experience the connection between sciences. Each group of 3 or 4 students gets a unique plant to study. Some plants are well known, such as clove and rosemary, and some are endemic or understudied. A large proportion of the plants we screen are native to California. Those plants we grow in the garden to minimize the impact on the environment. In the period of 4 weeks, students collect and dry plants, perform solvent extraction, and learn how to perform 3 phytochemicals assay to test for antimicrobial, anti-cancer, and stimulant or sedative properties of plants. You can find the details of those assays in the paper attached below. Students analyze those preliminary assay results, do some background literature search, and come up with original hypothesis-driven follow-up experiments that contain all necessary control and replicates to perform statistics. Some students are able to develop new bioassays that would explore other medicinal properties, an example of which will be highlighted in this poster. Let me introduce our student presenter, Aditi Poddar, who studied antimicrobial and antifungal properties of a curry plant.
Hi, my name is Aditi Poddar, and the plant that I further studied was Helichrysum italicum, or the curry plant. Our central finding was that Helichrysum italicum, or curry plant ethanol extract, has greater antimicrobial properties than other solvents. The other solvents we looked at were methanol, acetone, and hexane. Additionally, we tested all of our extracts on Pseudomonas putida and Staphylococcus aureus. This image shows our main disc diffusion assay. We assigned methanol as our negative control without any plant components added, and bactine as our positive control, of which we expected to see a large halo diameter. We also assigned 3 replicates of extract per plate. Our null hypothesis was that there would be no significant difference regarding the different extracts and the halo diameters we saw. And our alternative hypothesis was that there would be a significant difference between solvents. Essentially, plants are cheaper and have fewer side effects than standard antibiotic resistance. Our experiment focuses on the properties of the curry plant. Lots of previous literature shows that the curry plant has both antimicrobial and antifungal properties. The first step of our experiment was creating our extracts. We collected, dried, and grinded our curry plant into a fine powder and added 1 milliliter of 100% of our solvents, methanol, ethanol, acetone, and hexane, with about a 1-to-1 volume-to-volume ratio. We let this steep for 15 minutes, vortexed it, and repeated this one more time. Finally, we microcentrifuged our tubes at maximum speed for 1 minute. For our disc diffusion assays, we swabbed TSA plates with either Pseudomonas putida or Staphylococcus aureus. And we divided each plate into 5 sections for our positive control, bactine, negative control, methanol, and 3 test sections. We also added filter paper discs in the center of each section after we added 10 microliters of either extract, our positive control, or our negative control to the paper disc. And we expected to see a zone of inhibition around the disc. We incubated our plates for 24 hours at 30 degrees Celsius and moved it to the fridge afterwards. For our yeast growth assays, we obtained 9 jars and balloons. We heated up sugar water to let the yeast rise in the jars, and we added about 50 milliliters of water and 20 microliters of our extract to each jar at a 0.04% volume to volume ratio. We let the yeast rise and cover the top of the jar with the balloon to measure the diameter change throughout the 25 minutes. And throughout this process, the balloons were incubated at 30 degrees Celsius. We also subtracted the initial diameter of the balloon from the final diameter to see the total difference. Our results showed that the ethanol extract was more effective than acetone on Pseudomonas putida, and that the ethanol extract was more effective than both the acetone and hexane extracts on Staphylococcus aureus. We ran a one-way ANOVA test and a Tukey HSD for this data. The top image shows our balloon yeast assay exploring antifungal properties, and the bottom graph shows that the average balloon diameter increases over time for all samples. All extracts showed antimicrobial activity, which is supported by previous research. We see that the ethanol extract was more effective than acetone on Pseudomonas putida, and it was more effective than both acetone and hexane on Staphylococcus aureus. Our antifungal assay was successful, however, it does require increased extract concentration to see inhibition. According to Bacali, extracts had no effect on yeast growth when diluted to a 1:10 volume to volume ratio. So in the future, we plan to add a larger percentage of plant extracts into yeast reactions. We see that the curry plant ethanol extract is an effective antimicrobial agent, and further research will explore additional applications of this extract into other medical fields. I would like to thank Professor Tatiana Kuzmenko, Professor Demian Willett, my lab group partners, and the LMU biology department. Thank you.